All right, everybody, let's give a quick rundown review of chapter 15, which dealt with innate immunity. There was a whole bunch of lead-in into the structure of the innate immune system, the lymphatic system, and everything like that, which I'm not going to necessarily review here, but let's go over the newer, cooler, more detailed stuff here with the time that we have. So our first line of defense, that's going to be our physical and chemical barriers. That's going to be like the skin itself. That's going to be like the acidity of the stomach. That's going to be like, blinking. All of those things are considered part of the first line of defense. Okay, they are not related to the immune system, but they do give us protection. Lysozyme is present in like tears and saliva. That helps break down peptidoglycan, and it's important to kind of know the function of that one, FYI. It's an enzyme that breaks down peptidoglycan, which is part of the cell wall of bacteria. Useful enzyme. Also important, we talked about keratin, which is basically a compound that seals and waterproofs our skin so that it's more impermeable. So keratin is important. All right, the immune system itself. So that includes the second and the third line of defense. It has to be able to recognize self versus non-self, and that's going to be based on surface markers of our cells that we can recognize as being self-surface markers or non-self surface markers if you are an invader. The lymph system, so this is a little bit of a rundown of those parts, right? The thymus is involved in maturation of the T cells. The lymph nodes are important where we have interaction of our white cells, start activation of lymphocytes, and start the acquired immune response, which is in the next chapter. And then the spleen, which filters out blood, but not lymph necessarily. So remember, the lymph nodes are going to filter out lymph. The spleen is going to filter out blood. It removes damaged red blood cells, but it also will associate with like other lymphatic cells, whether that is associated with the lymphocytes or, activation of monocytes in response to these as, phagocytes. And all that. So it is considered a lymphatic tissue in addition to its primary role of removing damaged and dead red blood cells from your bloodstream. All right, and then the lymphatic fluid itself is just essentially the fluid portion of blood, plasma essentially, serum really, that has moved out of the bloodstream and into an inflamed area. And then it comes back out of that inflamed area and moves via the lymphatic system from the tissue towards the heart. Lymph does not move from the heart to the tissue. Blood does that. And then fluids move out of blood into these tissues and then come out as lymph fluid, and then move back towards the heart. As they do that, they will get filtered out by lymph tissues like the lymph nodes. Where you can interact with the pathogens and start activating appropriate responses. So that's essentially what is going on there. And that movement of that fluid is driven completely by muscular contraction. I'm not talking about smooth muscle. I'm not talking about your heart. I'm talking about, I'm not talking about cardiac muscle. I'm talking about moving your legs moves that fluid, and that's the only way it's going to move. So if you sit too long, you'll start collecting fluid in your tissues and it won't be able to move out. It's important to move. It's important to move, especially if you are healing. All right, so white blood cells are also known as leukocytes. There are some different kinds of leukocytes, white blood cells, granulocytes and agranulocytes. Granulocytes include the neutrophils, eosinophils, the basophils, and the mast cells. The most numerous ones are going to be your neutrophils. So they do have phagocytic action, in addition to the monocytes and the macrophages. Mast cells have it too, and the dendritic cells, but I'm not as concerned about the mast cells, guys. We're not as concerned about those. Okay, sorry. All right, my bad. Should click on something. Okay, so right, so we really want to be aware of the neutrophils, the monocytes, the macrophages, the dendritic cells. So those are some of our biggies as far as our phagocytic cells. A lot of these guys are going to be reacting to PAMPs. These are just pathogen-associated molecular patterns. I don't know what else to say about them. So those are just going to be things that we just automatically, in general, see as a sign of foreign, whether that is due to the presence of LPS, which is endotoxin, shouldn't be there, or peptidoglycan shouldn't be there, or double-stranded RNA shouldn't be there. None of those should be seen. So if we are seeing those are examples of PAMPs that we would just immediately respond to those. Those are not specific to a specific pathogen, the way that an antibody would be. All right, phagocytosis is eating something up and ingesting and breaking it down. Our steps were chemotaxis, which is movement to the area where, the pathogen is due to chemical signals from nearby cells. And ingestion, which is going to be literally engulfing it as a phagocyte. With the phagolysosome formation, when you eat up the foreign guy, you put it into something called a phagosome. It's like a little vacuole or package that allows you to just keep it in that little bubble. Now that'll merge with a lysosome, which contains lysozyme, as well as other molecules that will help you kill the invader that's inside. So it could be toxic oxygen molecules, it could be acids, it could be other, toxic substances produced by your cell in order to lead to killing of that thing. So that is our destruction step. And that only takes about 30 minutes, and then you will excrete what is left over from having killed the pathogen. That's how phagocytosis works and how it helps us with our invaders. So agranulocytes, those are granulocytes, neutrophils, fills. Okay, fills, it's the fills and the mast cells. The fills, though. The agranulocytes, now we're talking about our monocytes, which are phagocytic cells. Then if they move and differentiate into specific tissues, then they are macrophages. If they're mostly in the skin or in other areas as part of the mononuclear phagocyte system. So that's going to be the areas that allow these cells to move around between the tissues. All these guys are antigen presenting cells. They are antigen presenting cells that are going to help activate lymphocytes, the B and T cells later on. B and T cells are the lymphocytes. They're the only lymphocytes. Okay, those are the lymphocytes. Well, there are others, but those are the ones you need to worry about. Anyways, the agranulocytes that are involved in the innate immune response are going to be your macrophages, monocytes, and dendritic cells. However, they have quite the important role to play with regards to the acquired immune response, because they eat things up and present them to our acquired immune cells, our lymphocytes, and ask them, hey, doesn't this look like bad guy that you respond to? And they'll be like, yes, this does look like bad guy that I respond to. And it helps, activate that whole response. So they're very important in that response as well. All right, so the inflammatory response, when we're talking about the innate response, we're not talking about necessarily producing antibody. That's going to be the next step. So what does innate response do for us? The inflammatory response is what it does for us. So we have all sorts of chemical signals made by different cells that have us respond in different ways. But in general, the inflammatory response is going to include rubor, which is the redness due to the vasodilation, calor, which is the warmth due to increased blood flow to the area, tumor, which is swelling, which is due to the edema from the fluid into the area, and then pain due to the nerves being damaged and affected, and eventually loss of function in that tissue. The events that are involved in inflammation, we have essentially 4 steps. The injury reaction, which is like you get hurt, step on a nail, for example, which is what was the example given. So your cells immediately in that area will start reacting and responding and essentially sending out signals for increased response. So then we have the vascular reaction, which is the bringing of chemicals and cells that are involved in the inflammatory reaction to the area from the vasculature. That would include neutrophils. Then we have the next step, which is going to be the edema and the pus. So we have the fluid coming from out of the bloodstream into the area. That includes the chemicals, that includes the cells, that includes all of the different proteins that are going to be involved in healing the area and responding to the whole process, right? That's edema, so fluid into the areas and swelling. And then pus formation, which is going to be, all the neutrophils, other blood cells and dead tissue cells, and the bacteria all together. That's going to be the pus, as well as fluid in the area. And then we have healing and scarring as a result once we kick the whole infection. the healing and scarring, that kind of step in between, as far as the edema and the pus, and then the healing and scarring. In there, we have introduction of the acquired immune response, or the specialized immune response from the B and T cells. So they do come in there and sort of specifically attack invaders and help get rid of them very specifically, and therefore develop a response, a memory reaction that is specific to those organisms, so that you can, if you see them again later, you're protected. You won't even know that you were exposed. So white blood cells will move into tissues and respond to those signals. They'll move into the tissues via something called diapedesis. That's their ability, it's a very unique ability to move between the endothelial cells that constitute the blood vessels, essentially, to get into the tissues. And so they can migrate into those tissues and respond appropriately. And as far as these mononuclear cells are concerned, they can associate and move in between the tissues using that mononuclear phagocyte system of the whole connective tissue in between, so they can get between the cells and do their job. Fever. Fever is induced by pyrogens. It basically triggers the hypothalamus, which regulates your temperature so that you would have a higher temperature. And depending on how high the temperature is, you may or may not choose to treat it. Use your judgment, and of course, consult your doctor when you're not sure. But essentially, fever itself, it increases your metabolism of your own cells so they can start fighting back faster and sooner. And it creates a protective response that will be increased due to that temperature as well. Then we have decreased microbial enzyme productions, which will decrease the microbial growth, and they can't use, the virus. They can't use iron from, the system well, so that inhibits their growth as well. All this stuff that helps increase our activity and decrease pathogen activity. That's the point of a fever. Fevers can be caused, like we said, they're caused by pyrogens. Pyrogens can come from an outside source or an inside source. Outside sources are exogenous sources. So that would be like from an invasive virus or a molecular marker that would be associated with an actual, like a bacterium that's causing an infection as a pathogen in your body. Endogenous sources from inside your body, those are like chemical signals from the cells to one another. There are also chemical signals that can trigger the development of fevers as a protective factor. All right. Interferon is a little protein that we have that's made by cells that will allow the cytokine. So that's a little protein made by cells to signal to one another. But basically it signals nearby cells that there is a viral infection or even cancer that can trigger it as well. It says that I'm being invaded by virus, and it lets all the nearby cells, no matter what kind of cell they are, they can get that signal and say, oh, you're getting invaded by virus. I'm going to go ahead and start making antivirus, essentially. So that's a watered down version, but that's the point of interferon. It allows us to signal and help with any internal infections like that. All right, complement cascade. We introduced the concept of complement, which is one of the coolest things that your body can do. The steps involved: initiation, amplification, polymerization, and membrane attack. Initiation is literally getting signaled by the presence of antibody or sugar, or often mannose via lectin, or LPS, lipopolysaccharide, which is endotoxin essentially. So antibody is classical. Lectin is the lectin pathway and recognizes sugars on bacteria, special sugars. And then alternative pathway is going to recognize LPS or endotoxin. So we've triggered it that way. That's our initiation. We amplify by, basically we recognized that there's this antibody, there's this, lectin, there's this LPS. Now our protein in the complement cascade, with those 50 potential proteins that are floating around in the bloodstream for complement, they're going to get triggered and activated, and then essentially cause a cascade of changes within the proteins that are involved in the whole system down the way, and activating one another through stepwise changes. That can lead to opsonization, which is helping out phagocytosis essentially. cytolysis or inflammation. So inflammation, we've just talked about. Cytolysis, though, here we're talking about the membrane attack complex that is generated by complement and just pokes holes in the target pathogens and affected cells to allow for us to destroy them that way. So that's what complement can do for us. And that's pretty cool stuff. So that's it for chapter 15, and I will see you guys for the next one.